However, the cancer management, because of the diagnosis and advent of new therapies and the either we call it AI or just call it intelligence, because we are able to use existing therapy better, I think we are making enormous progress. But how and where we are going is that diagnosis bit early, identifying the driver mutation, looking during the treatment how patient is responding, and combination of those three is the way to, I think we are making progress in that direction. Yeah, so hello everyone. My name is Mukesh Nyati. I'm a professor in Radiation Oncology. By training, I am a biologist. I have trained a little bit in medicine chemistry, primarily in biology, in signal transduction. I joined University of Michigan in '98 as a postdoc, and then I stayed here. So this is my 25th or 26th year here in the campus.
Oh, nice. So thank you. Well, you're currently working on mutant epidermal growth factor receptor demertization inhibitors.
let me say it again. Yeah.
I probably butchered the pronunciation.
It's okay. It's all right.
But,
yeah, I'm working on EGFR protein. Yeah. So what is the question?
And the question would be, I mean, you got a series of several large grants to work on this inhibitor. Mm-hmm. What's the progress on the inhibitor, and what's the impact on patients if you succeed in treating this?
Yeah, it's a good question, and even I need to figure out the right answer someday. But to address first question, what is the progress? So I started this project in its idea 2007. And as you know, drug development is very, very hard. It takes several $1 billion and many years to be successful. So I started in 2007 to understand receptor behavior that we can discuss later. But in terms of progress, I think we are ready to start IND enabling talks, which means we are ready to do one more step before we enter in clinical trials. So now we are soliciting different CROs. These are contract research organizations that they can do safety studies for us in mouse and in dogs, so that if we find that a safety profile is all right, then we can enter in clinical trials. So that's where we are in terms of progress. And I guess what was your second question?
It was, what's the impact on
What is the impact? So
patients?
I think you have to then take a few steps back in terms of what exactly I'm trying to do. So for your audience, which may or may not be all scientists, so maybe I'll say this way, that cancer is a very complicated disease. Scientists have been trying to treat cancer for 100 years or more, and we are successful in treating many different types of cancers, quite a bit. But as we all know, that there are different type of people have different cancers, and sometimes it is very complicated to treat patients. So there are a couple of genes or proteins that are very important in large majority of cancer patients. One of them is EGFR, which is epidermal growth factor receptor. And one of the other very important molecule is called KRAS. These two molecules are generally present in their wild type form in most tumors or solid tumors, but in some cases they are mutated. Now, whether they're in wild type or mutated form, their primary function is to give a signal to cell to start proliferating or dividing. And that's what cancer is. The cancer cell actually divide for no function, no reason. So what I have been trying to do is to target these two proteins directly or indirectly, but in a way that has not been done before. So if I'm really successful, I means here when I say I, it's entire team work, but if this project is or we are successful, then we may actually do four things. One, we may actually start a new line of therapy, completely novel treatment. And if those patients really respond the way our mice does, then I think we'll be able to help many patients who are currently not being targeted by any of the existing drugs.
So that would be a therapy separate from chemotherapy?
Yes.
It would be a drug, a pill?
Most likely it will be a pill. But chemotherapy does not mean it's not a pill. Chemotherapy can be a pill, chemotherapy can be given IV, and so on and so forth. Chemotherapy simply means a therapy involving chemical. So this is also chemical, but it will not be classically considered a chemotherapy. It will be considered personalized medicine. but it is similar to chemotherapy. And yes, we envision for now it will be a pill.
And it would be a new therapy, so it would be helping cure cancer. That's really great.
That's the hope, yeah.
Fantastic. So on cancer research, so you've been doing this for 3 decades.
Yes.
You've been studying cancer. Could you tell us a little bit more about how your focus through those 3 decades has changed over cancer, over your research into cancer, and really how that's evolved over time?
Sure. So first of all, I would say my focus has not changed. For last 30 years, I'm trying to do the same thing as I started as a undergrad or PhD student. I'm still trying to do the same thing, but the focus just evolved rather than change. So earlier I was trying to understand how existing chemotherapeutics works. Then I started to work on gene therapy project where I wanted to deliver chemotherapy at very high concentration or dose to a particular tissue. And from there, I began to, as personalized medicine became very popular in early 2000, one of the research articles I read in 2001 was about Herceptin, which is a drug that blocks HER2 in breast cancer. And when clinical data came quite positive in 2001, it was I think it provoked some sense in me or idea that maybe I should work in personalized medicine. So I started to study HER2, and I found that HER2, which is mostly expressed in breast cancer, it has 3 other sister or family member. EGFR is one of them. So in 2001, I started to read about EGFR. And in Pfizer, they were developing a drug called CI-1033 here in Ann Arbor. So I actually got a project from Pfizer to research their molecule in 2001 as a postdoc. During next 5 or 6 years, I published few articles. This drug, or the CI-1033, was never approved because of toxicity and maybe some business decision. However, I learned few things, and my primary thing that I learned during that time was that EGFR can be targeted by CI-1033, and the inhibition of EGFR activity does not correlate with how cells respond. So I start to research why many cell lines or many cell types do not respond to CI-1033, in spite of the fact that they actually overexpress this protein called EGFR. And in 2007, I noticed that it is actually EGFR protein itself, in addition to its kinase activity, that is very important for its survival. So then my goal shifted to figure out a new therapeutics that could target EGFR protein by removing it from the cell rather than just inhibiting. So next 2007 till 2024, I'm working on that area. So that's how my, idea evolved or shaped.
So removing it from the cell instead of inhibiting.
Right.
So I'm not a technical expert in this, so can you tell us a little bit more about how those 2 approaches are different from one another?
Of course. One simple example you can imagine, having your money in your pocket will be kinase activity. So you have money, you have kinase activity. You have money, you can go and spend, buy stuff. When you have kinase activity, most of the time, that's a signal for cell to divide. Okay? But removing it is essentially taking you out from the market side, right? Not money, but you don't exist anymore. Right? Or taking your wallet away or taking your credit card away. So when you remove a protein selectively, that is very important thing, then whether it has a kinase activity or not, both of the function can be gone or will be compromised. So imagine it this way, that I take your allowances or money out, but you can still earn money tomorrow. So what happens in the cell when you inhibit kinase activity? Yes, there is a response. There could be dramatic response in certain tissue type or certain cancer type, but most of the time cancer cells figure out how to get that kinase activity back. So the effects are either short term or cell can mutate again and then drug become ineffective. However, the idea was or is, if you remove the protein, then you cannot mutate it, right? If the protein is not there, you cannot mutate it. And that was my idea, that if I can selectively remove EGFR from those cells that depends on EGFR for their survival, we will have a better chance. Now, having said that the idea is very easy or cheap. Data is very expensive, because how to really selectively remove a protein from cancer cell is not a joke. So that's what I researched in next 15 or 17 years.
This is something that's a huge deal in the medical industry, that people have been trying to crack this problem, and it's really as you said before, a set of many different, very difficult problems. What has been the story of cancer research in the past, several, really hundreds of years that we've been trying to crack this problem? And where do you think we are in that story of cancer research and development?
I mean, it's very hard to describe in few minutes, but we started cancer therapy from a standpoint of radiation or surgery. This is how actually things started. And after that, it was certain drugs like nitrogen busters, the alkylating agent. These are actually poisons. But this is how the field of chemotherapy initially evolved. But I think in late 85 or 90s, people realized that you cannot just use generic chemotherapy, but you have to understand how the chemotherapy works and maybe develop drugs that will work for certain patient. At least you know that thyroid cancer is different than pancreatic cancer. Certain therapy works there, certain do not work there. And then this is where the personalized medicine started. It also started because we understood a little bit more about genomics. So certain tumor has more mutation, some patients do not have those mutations, etc. How we are moving is, as you can actually see, the cancer is still devastating, right? Every time somebody is diagnosed in your family or friend circle, you do have a long phase. You do feel a little sad that what's going to happen. However, you do know there are certain type of cancer, like there is a lymphoma called Burkitt's lymphoma. It's not very common here in America, but in Africa and children, it's a quite common disease. But Burkitt's lymphoma is almost, I would say, curable. Like that, thyroid cancer is fairly manageable. Same way as the prostate cancer, at least in 90 to 94% patients, prostate cancer can be managed. Same as breast cancer, if HER2 is positive, Herceptin really works. And like that, there are a bunch of examples. So personalized medicine using specific molecule that target specific proteins, etc., do work in some patients. But where we are moving now is going further deeper, really identifying how patient responds during therapy. So when a round of chemotherapy or personalized medicine is given, many times patient samples are collected, whether they are liquid. So you are just looking at the circulating DNA or looking at the normal tissue response or looking at the tumor house cells responding to the therapy and then adapting therapy while the treatment is actually not completed, just to figure out if the patient is responding or not responding. So that is one way, is just to fine tuning the existing treatment and finally and using treatment in a more, I would say, logical manner. Right? That is one area. Then other area is support system. When patients are treated, the toxins are more well managed these days compared to past. So people can actually handle more therapy, longer therapy, or higher doses. And then third area is immune therapy. That is now quite prevalent. Certain patients, certain tumor type do respond to this immune checkpoint inhibitor quite well. Now, whether immune checkpoint inhibitors are synthesized in lab or they are also done where patient cells are taken out from the system and then they are remodified and re-injected into the same patient. So those kind of therapy also works very well. Gene therapy is another way, protects are another way, molecular glues are another way. So the field is evolving very rapidly, and as you know that AI is everywhere, whether you use ChatGPT or you use in drug development. So the information that we are generating is enormous, and using the information to fine tune our therapy, find the right doses, right, or find the right therapies is the way to move forward.
So thanks for those explanations. And so while Eric was talking about the history or current stage, or where do you see cancer therapy, or maybe the research you're working on, where do you see it go forward?
Oh, I think I just said the same thing, but yeah, I think we are making enormous progress. It will, in my opinion, we will not have for example, what we noticed with COVID. It's almost now, you can say in a non-scientific language that COVID is gone. It is not true, but you can say that it is quite well managed, right? Same is for HIV. We cannot say this for cancer. However, the cancer management, because of the diagnosis and advent of new therapies and either we call it AI or just call it intelligence, because we are able to use existing therapy better, I think we are making enormous progress. But how and where we are going is that diagnosis a bit early, identifying the driver mutation, looking during the treatment how patient is responding, and combination of those three is the way to, I think we are making progress in that direction.
So on that, where you mentioned the patient and seeing how they're reacting to certain doses and therapies. Do you think that it would be a one-size-fits-all
No.
solution, or do you think it would be no? Do you think it would be personalized?
Yes, it will be highly personalized. Personalized medicine is overused term, but I would say it will be truly personalized medicine as we move forward, because your entire tumor can be sequenced very easily these days. You can figure out what mutations, 1, 2, 3, what kind of mutations you have, and based on the mutation and based on the profile of so many drugs that we have, and we know how the particular drug or particular agent works in which tumor type, those kind of things will come into play much greater role than currently we have in our hand. So no, it will be highly personalized medicine. We are making progress, and that's how we are, I think, going to go into future.
I can't wait for for the next developments.
Sure.
I think that's going to be truly something that will be listed in the great accomplishments. It's good work we do. With the role of artificial intelligence and sort of using advanced computational methods for cancer research, what computational methods do you think would really level up cancer research?
I think it's not just about cancer. In any research, in any field, you can think it's the data. As the more data is available and high quality data is available, you do need this kind of algorithm to figure out very quickly where we are, because you may have data already. You see that how many thousands and thousands of patients have been sequenced and the data is available, but in order to read the data quickly, right? Same way there are thousands and thousands of preclinical development program exists in the country or in the world, but we need this AI to see patient, their genomics, proteomics, etc., and also the drugs that works already, and then how to figure out which drug may work in what combination, right? In what schedule, how much, for how long. Those kind of questions right now are subject to illusions. Some brilliant mind can figure out, but I think with the AI, I hope it will be better for all.
Yeah, that's one thing I wanted to ask you about and to get your opinion on. I recently listened to a podcast with Sam Altman, and he was talking about how they use large language models in research environments to, and he gave the example, cure cancer. How long before we have a system that works with because you lead a lab of multiple students, how long before it's in the lab testing experiments, automatically controlling the lab equipment to run experiments, collect data, form a hypothesis, and test that hypothesis?
Oh, I mean, that will be more of a, I think, fiction movie.
Yeah.
I do not know whether we need to be there or whether we will ever be there, but it's very hard to predict about future. So I cannot put a timeline and when that might happen. The question is, do we need that? I don't know whether we need to have that level of automation, but already where we are now, we are able to see, as I mentioned before, lots of data that how a particular type of cancer evolves over, let's say, a certain period of time, a month to 10 years, right? And what happens to those patients and what kind of therapy really works or does not work. Because sometimes it is also very important to know what does not work so that you don't try those therapies unnecessarily, right? Because then you're wasting the time, and the time is the essence for everything. So directly I cannot address your question, what time or when that may happen, but in some extent it is already happening. There are so many instruments even here in the same building where somebody can actually handle the instrument from home. You know, using some sort of gaming device, you can adapt it, and we can manipulate some of the robotics. So it is happening, but to the level where it will generate new hypotheses, I'm not sure, because that's where you do need a little bit discrimination. The artificial intelligence, you still have to remove the artificial part, and intelligence is still will prevail. So I think you do need human who is watching all those things, making sense of the data, writing new algorithm as the more data become available. Because sometimes all data does not mean it's a good data. But who does the curation? That what is the good versus not so good data? And that's where I think it's going to be a combination of people who are now getting trainings in artificial intelligence. At the same time, people who go deep and dig deeper and understand what to look for, when to look for, how much to look for, how much to depend on all this algorithm.
That's
But I think we are moving in this direction, and probably it is not going to be binary. I think it's going to be bidirectional. Statistical genetics means essentially you're just looking at the genomics of whatever, right? Cells to animals to human to different disease. And that's an extremely important, very valuable data set. I would say that's the first stage or first step. That's where we start from. We have to understand the genetics of anything, how it is normal and what is not normal. So once you understand the normal of a given disease type, right? Not just cancer, for anything for that matter. Once you understand normal and then you know what is not that normal and what are those factors, and that's where the statistics genetics comes in picture, and followed by what medicines already exist, and then how can you develop or should we develop new medicine. I personally believe that we have enough medicines already, and it's not like when I say enough means it's a binary. There is nothing binary, right? But we already have very effective medicine, but sometimes we don't know how to use them. Also, sometimes we are very late in using them, right? And all the medicines are not available throughout the world. So diagnosis is one other part. So genetics and statistics will continue to play very important role in diagnosis, followed by finding the right type of combinations or therapeutics. And then third is to figure out very quickly or in the middle of clinical trial or treatment period, what is working from what is not working, so that we can adapt the therapy for those where therapy is not working. So at all four stages, I think the genetics does play a very important role. And so is the statistics.
Yeah, so you lead a lab, as we mentioned earlier, of how many?
So at any given time, I have only 4 to 5 students.
4 to 5 students?
Yes.
And you also collaborate, I assume,
Yes.
with other researchers. What's the role of collaboration?
Oh, what they say, like, it takes a village to raise a child, right? I always say that in my talks because my acknowledgement slide is the busiest one. It's actually full page of people's name because no one alone can do anything the way we are moving forward. Gone are those days where 1 or 2 authors publications were prevalent. Now it is 10, 20, 30, 40 authors and then followed by acknowledgement sections. So what is the role? Role is, it cannot be put in right words other than that you alone cannot do anything. You do need a team. You need a right team. And team includes people as well as infrastructure, as well as funding, coaching, mentorship, and students. So it takes a whole 9 yards to do anything. In my case also, it is true. So I have been so privileged to work with my mentor, with who I did my postdoc. He's my now collaborator. He's MD PhD, so brings a experience from what happens in clinic. I have collaborators who are computational chemists, who computes, who figure out using the existing information that what and where we should go. Then I have a medicinal chemist who synthesize molecules based on what works or what did not work. And then I have a OTT, Office of Technology Transfer folks, who actually have I would say quite regular discussion on our discoveries so that they can review whether this discovery is worth patenting or not worth. And then I have business partners who actually help in understanding or giving the perspective from the market side. What is being developed, what are the things in pipeline, and where the IP and where we are, so that we can actually use our time appropriately. And it is also very important to have, let's say, grant support. So in my department, we have a extremely good support for funding or writing our grants. And again, it's a very complex, I would say, and complicated part of the work that we do, where a scientist normally cannot do those kind of paperwork accounting. So collaboration is at multiple levels, at intellectual level as well as the infrastructure level. And then I, personally speaking, do not work on bench. So I need students who actually take their time off, come, after classes or before classes. Sometimes they work overnight or 24/7, I would say, depending on their interest or their enthusiasm. And that's how we make any progress for that matter. So collaboration is the key.
Wow, that's fascinating. All the different roles that go into making this, and I had no clue there was so much.
Yeah, and also it's not just the people and infrastructure, but the duration also. The perseverance of not just you, but many people around you. Because in drug development, I would say there are only few good days in your life when you are doing a drug development, and those good days could be that I this was my hypothesis number 10, and finally I have something. Most of the time you have only nothing but disappointments, because you can predict whatever you want, right? Or whatever data suggests. But when you really execute experiments, many times the data do not fall as predicted. So then what happens? Then you need a support system where you review and look at the same data from 3 or 4 or 5 different perspectives and figure out what is truth and what is not truth. And there, that mental support is also very crucial as you make progress. So collaboration is the key. Without collaboration, I would say, in this stage, nobody will, or I don't know who will progress.
Yeah. So it's gotten so dynamic and so complex
So complex.
that not a single person can do this.
Nobody can do. And also complex, and then it's probably not a right flavor for me to come this way, but there is a high competition also, right? So meaning if you are doing by yourself, it is possible that you may achieve what you want to achieve, but it will take you 10-fold more time. By the time you achieve what you wanted to, you are already too late. So the timing is also very valuable, and therefore you do need a very good team that supports you, stay with you in thick and thin, and also guide you. Sometimes you also hit a stone wall, and if you cannot make a hole in the wall, you have to return. You have to find a different thing to work on. After you have given enough efforts or attempt, then you have to sometimes say, I'm done with this. So for that call also you need collaborators. Something like reality check, right?
Yeah. So on the collaborator front, so there's multiple different roles, and on the mentor front, and on the great scientists that you really look up to and try and emulate in your own, who are some of those people?
And the list is very long, but if I have to just put a couple of names, I would say Ted Lawrence, who was the chair of Department of Radiation Oncology. He is my mentor. And another scientist was also MD, Bill Brett, who taught me a lot of lesson in biochemistry at the University of Michigan. These are the two names I will just... And what did I really learn from them? I think is two things. One is perseverance, and second is honesty. The idea is, what I think I learned from them is follow the data, nothing else. Not even your own hypothesis. You can make hypothesis, you may have to redo your hypothesis, but you must follow the data and stay honest to your data. So those are the two mentors I would say has given me enormous help to shape my ideas.
Yeah, I mean, everybody's got mentors, everybody has people that they look up to.
Yeah, photography is my passion, just like science. And I see they are parallel in the sense that if you want to be a good photographer, not just looking at a subject and click the same angle over and over again, those pictures are not useful. Same way in the research, you can look at the same data or same sort of profile or same sort of problem, and unless you have a novel angle to look at the problem or novel angle to look at the picture, what do you want to capture, right? So attention to the detail, and let's say looking from a different vantage point, different angle. And that's how I draw a parallel between my science, the way I do, and the photography. You have to be different. You have to have your own picture. So if I click a picture, I call it my picture. It is not the picture of the subject, or not the picture of the landscape, not the picture of the sunrise or sunset. It is my picture. Same way when you write a research article, it is my article. Mine not in terms of the egoistic mine, but mine is that I or my team contributed towards that science. So it's attention to detail, novelty, looking differently. And another thing is that you need patience, even for photography. You cannot just click picture or pictures unless your mind is completely calm and still. And you have to wait for the right time. If you're looking for solar eclipse, if you miss one, you have to wait for another year or 4 years or 10 years. Same way, unless you are very calm and composed in science, you cannot achieve, I would say, any success. So patience is the key for the photography as well as the science. That's how I draw parallel between those two aspects.
I also noticed that you're a big fan of nature, and
Yes.
I saw some of your photographs, and they're just phenomenal.
Thank you.
I must say. And I noticed the common theme among them was nature.
Yeah.
And what do you think is so mysterious or beautiful about nature that we see?
Well, I mean, that's a topic that probably we should talk an hour on that, or maybe more. But we are part of nature. We are not separate, right? So We can see us as extension of the nature, or we can reverse the thought and say that nature is extension of me, right? And every time I look out, whether it is somebody can call it bead, or you can just be fascinated with how the spores are germinating and with how things are developing and how insects are behaving with their circumstance, with their environment, and everything that you see around you is fascinating. You just need a little patience and time to look at it. You just have to slow down a little. So nature is indeed fascinating, whether it is rising of a sun or moon. It fascinates me every day whenever I see it. Or a bumblebee or even a hornet. When you look at them, it's just beautiful. Extremely intricate, complex, even mosquito if you think of that. Think of ladybug. Whatever animal you look, whatever organism, plant, sand, water, or science, it's just beauteous. It's actually mysterious. We think we know, and perhaps we know very little. And that's what it keep me going and keep me allowing to take my camera pretty much every day out and do something with it.
Wow, that's really beautiful and humbling as well. And in the fact that, I mean, you as a cancer researcher and as a photographer, you see both the beauty and greatness of nature, but also the capacity to destroy our body and cancer.
Yeah.
So
It's a very fascinating actually. If you slow down a little bit and you will start looking, then you will find there is no bad day. It's always a great day, right? It's just perspective. So nature keeps you, I guess, humble, gives you energy to do what you do every day and continue with that. Yeah.
great. Well, I think we have a great interview, and I want to thank you so much for coming in, sharing a bit of perspective into your world, the world into oncology research, and yeah, thank you so much for
I appreciate your time. I appreciate those questions. I also had fun. Hope you find something useful and hope your readers or audience find something to cheer.
I'm sure there's